Logo, NIH National Institute on Aging. Text, https://www.nia.nih.gov/SBIR slide. The webinar will begin shortly. Questions and comments. Share questions and comments in the chat window in the right side of your screen. Questions will be reviewed during the Q&A period. Closed captioning. http://bit.ly/2dnix8p Zoom technical support. +1 888 799 9666 extension 2 or https://support.zoom.us/hc/en-us.
Thank you for joining today. The webinar will begin in about 1 minute. We invite you to take a look at the closed captioning and technical support. Thank you everyone for joining the webinar today, Tapping into Small Business and Biotech Funding to Accelerate Alzheimer's Research Innovations. Throughout the webinar, you can submit questions and comments. To share questions, please look in the chat window on the right side of your screen. Questions will be reviewed during the Q&A period. We also have closed captioning available and technical support. If you have any questions on technical support, please call 888-799-9666. Please know that we will have a Q&A session and that you can chat us throughout the webinar. This is an interactive opportunity.
Slide. Tapping into small business and biotech funding to accelerate Alzheimer's research innovations. Thursday, December 12, 2019.
I want to acknowledge our host today, the National Institute on Aging, as well as ADDF.
Slide. The National Institute on Aging, NIA, leads a broad scientific effort to understand the nature of aging and to extend the healthy active years of life. NIA provides more than $100 million in set-aside funding for the Small Business Innovation Research (SBIR) and Small Business Technology Transfer (STTR) programs. The institute offers dilutive grants to commercialize interventions that address aging, aging-related diseases, and the special needs of older Americans. NIA is the primary federal agency leading research on Alzheimer's disease, AD, and AD-related dementias, ADRD.
They are coming together to try to accelerate innovation.
The mission of the ADDF is to rapidly accelerate the discovery of drugs to prevent, treat, and cure Alzheimer's disease. The ADDF follows a venture philanthropy model, funding breakthrough research in academia and the biotech industry. The ADDF has awarded more than $130 million to fund over 600 Alzheimer's drug discovery programs and clinical trials in 19 countries. 20% of the drugs in clinical development for Alzheimer's disease received support from the ADDF.
Our featured speakers are Todd Haim. He's a director of the Office of Small Business Research at the National Institute on Aging at NIH. We also have Zane Martin. She's going to be answering your questions throughout the Q&A session of this webinar today. From ADDF, we have Alessio Travaglia. He's going to be talking about some new programs and ways for you to find out about funding. And I will be hosting the Q&A session of today's session. You'll be hearing from the National Institute on Aging, Office of Small Business Research, followed by ADDF, and then there will be a moderated Q&A. This is just a reminder, please do send us questions throughout the webinar. You can look at the right-hand side of your screen and hit the chat. We'll also collect feedback from you at the end of the webinar, so look out for a brief opportunity to answer some questions. With that said, I'd like to begin and turn it over to Dr. Todd Haim.
Slide. Agenda. Introduction and objectives. NIA's Office of Small Business Research. Program overview. Opportunities and resources. Alzheimer's Drug Discovery Foundation. Program overview. Opportunities and resources. Moderated Q&A. Please use the Q&A function on your viewer dashboard to submit questions at any time during the presentation.
Thank you very much, Monique. So I'll be talking about the NIA Office of Small Business Research and the types of programs that we have available. Next slide.
Slide. About SBIR and STTR. Fiscal year 2019 set aside 3.2% for Small Business Innovation Research SBIR program. Set aside program for small businesses to engage in federal R&D with potential for commercialization. Fiscal year 2019 set aside 0.45% for Small Business Technology Transfer STTR program, set aside program to facilitate cooperative R&D between small businesses and U.S. research institutions with potential for commercialization.
So for those that don't know, the SBIR and STTR program are congressionally mandated programs that serve as a vehicle for agencies that provide extramural funding, usually thought of as funding to academic centers and institutions, that a portion of those funds would actually go to small businesses. And there's two sister programs, and I'll go over the differences of both. Together, it's 3.65% of the extramural funds at each agency goes to small businesses through these programs. So each agency uses it as a way that best meets their mission. So a DoD, for an example, they would use it to fund the development of warfighter technologies that they could acquire. At the NIH, we don't use it to fund development of technologies that we would acquire, but rather to fund the development of technologies that help meet our mission of improving health. Thus, at the NIA, we fund the development and further development of technologies and research that maybe NIH previously funded through other grant mechanisms, but now to try to fund the development steps that are needed to advance a project further towards commercialization. Next slide.
Slide. Why seek SBIR/STTR funding? Provide seed funding for innovative technology development. Not alone. No repayment required. No impact on stock or shares, non-dilutive. Small business retains intellectual property rights. Provides recognition, verification, and visibility. Helps attract additional funding or support, e.g., venture capital, strategic partner.
There are a lot of advantages to small business funding through SBIR and STTR. The first and foremost is it is non-dilutive, which means we do not take any equity. There are no royalties. It is not a loan, so there is no repayment. Importantly, the small business does retain all intellectual property rights. It has been shown to provide recognition, verification, and visibility. A lot of outside investors like to see the validation that comes from the NIH peer review, and thus the combination of that validation and the data that can be collected in a non-dilutive fashion both help to attract the additional private funding that will almost certainly be needed to commercialize your technologies. Next slide.
Slide. Eligibility. Applicant must be a small business. Organized for-profit U.S. business. 500 or fewer employees, including affiliates. More than 50% U.S. owned by individuals, and independently operated or more than 50% owned and controlled by another. 1. Business concern that is more than 50% owned and controlled by one or more individuals. Or, SBIR only. More than 50% owned by multiple venture capital operating companies, hedge funds, private equity firms, or any combination of these.
In order to be eligible for the program, you have to meet the U.S. Small Business Administration's definition of a small business, which is essentially that you are a U.S. small business. You're owned and operated by individuals, citizens, or permanent residents of the U.S. And you can be owned and operated by venture firms now as long as no one venture capital operating company owns a majority of your company, of your small business. But if it's if multiple venture capital operating companies together as a syndicate own the majority of your business, then that would be now okay. Next slide.
Slide. Critical differences. A bubble above a table reads, award is still made to the small business. A table shows two lists under SBIR and STTR. SBIR permits research institution partners, e.g., universities. Small business concern may outsource roughly 33% of Phase 1 activities and 50% of Phase 2 activities. Eligibility. The PDPI's primary employment, i.e., more than 50%, must be with the small business for the duration of the project period. STTR requires research institution partners, e.g., universities. At least 40% of the work should be conducted by the for-profit small business and at least 30% by a non-profit U.S. research institution. Eligibility. IP agreement providing necessary IP rights to the small business in order to carry out follow-on R&D and commercialization. PI primary employment not stipulated, at least 10% effort to project. In separate box.
So as I mentioned, there are a few differences between SBIR and STTR. Important to note is that the award is always made to the small business, not to the academic research institution. In both SBIR and STTR, we do find that there is usually a partnership with an academic institution, although it is not required. However, with STTR, it is required. There are different percentages in terms of how much of the cost can go to subcontracts versus the small business. So for SBIR, 2/3 of the work in Phase 1 must be done by the small business, meaning 2/3 of the cost should be at the small business. Only 1/3 can be subcontracted to an academic institution. There are allowances for fee-for-service, which I can talk about. For STTR, you can have there's a minimum of 40% of the small business, a minimum of 30% at the U.S. research institution, and then the remaining 30% can be either of those two or a third partner. The other key difference is that for STTR, the PI can be at the university. For SBIR, there has to be a PI that's at the small business, but you could have a multi-PI for SBIR where one of the PIs is at the small business and one of them is at the academic institution. Generally, we see STTR as the model for companies that are really brand new spin-outs of the university, and that much of the work and leadership will still be done by the university personnel. Next slide.
Slide. SBIR and STTR program phases and funding levels. Phase 1, Discovery and Feasibility. Up to 1 year. Awards up to $300,000 or up to $450,000 for ADDF. Establish technical merit, feasibility, and potential for commercialization. Phase 2, Development and full R&D 2 years. Awards up to $2 million or up to $2.5 million for ADDF. Continues Phase 1 R&D efforts. Requires a commercialization plan. Fast track. 1 combined application for Phases 1 and 2 SBIR only. Direct out to Phase 2 SBIR only. Apply directly for Phase 2 funding, SBIR only. Demonstrated feasibility through other funding sources. Commercialization readiness pilot. Funding for late-stage R&D and technical assistance for commercialization. Phase 2B, competing renewal. 3 years. Awards up to $3 million.
So you'll hear many different budget numbers thrown out around SBIR, and it can be pretty confusing. There are congressional guidelines that are government-wide, and in the legislation, it states that the NIH is entitled to go 50% above those guidelines, so what they call hard caps. However, the NIH has successfully received waivers to go even above those hard caps for a set of topics that is really quite broad and expansive. So I think for many of the people, if not almost all the people on the line, you will be eligible to go above those caps, and in the solicitation there is a link to the set of waiver topics. If you can go above the cap, what we do in each solicitation is we state what the budget limit above the cap is for that solicitation. So for Phase 1, for most of our solicitations, it's up to $300,000. We do have a very open-ended Alzheimer's disease solicitation, PS 19-316. So anything that goes through that solicitation can be up to $450,000. For Phase 2, generally, it's usually $2 million at NIA, but for that Alzheimer's disease solicitation, it can be up to $2.5 million. Remember that Congress uses the phase nomenclature for SBIR, but it has no correlation with the clinical trial nomenclature, so Phase 1 SBIR is often preclinical, but does not have to be. There is also the ability to apply for Phase 1 and Phase 2 at the same time as part of one combined application, and that's called the fast track. The advantage there is that once you complete the Phase 1 portion, you will have to have set milestones in the application, and when you complete that Phase 1 portion, you would send us a document demonstrating that you have met all the milestones. And if we agree that the milestones have been met, then you would transition to Phase 2 without having to reapply and wait for review of that Phase 2 portion. For companies that already have proof of concept data, so as an example for a therapeutic, if you have in vivo data in a relevant animal model, then you can come in directly for Phase 2 now. And you just must show in your application what that proof of concept Phase 1 equivalent data is. And then you can come in straight for these Phase 2 levels of $2.5 million in Alzheimer's disease. We are happy to announce that we now also we accept the commercialization readiness pilot applications, and these are actually an additional $3.3 million. And you can apply either during your Phase 2 or after your Phase 2, and it can pay for late-stage R&D such as IND-enabling studies, manufacturing, scale-up, as well as technical assistance. And then after that, you can also apply for the Phase 2, or in parallel, you can also apply for the Phase 2B, which can be an additional $3 million. Next slide.
Slide. NIA OSBR core activities. Central coordination. Administer all the SBIR STTR awards at the NIA guidance. Help applicants prepare for application, resubmission, and discuss funding options. Outreach. Attend conferences and workshops and visit regional-based organizations to raise awareness of the program. Funding. Seed emerging technology areas by developing targeted funding opportunities and omnibus interest topics. Networking. Facilitate connections between awardees and potential strategic partners. NIA programs external partners. Entrepreneurship. Provide entrepreneurship training, as well as webinars on key commercialization-related topics.
So, I manage the Office of Small Business Research at the National Institute of Aging, And in our office, we have 6 key function areas. The first is to coordinate the program across the institute. We provide guidance to applicants, which I'll talk about. We are very happy to guide you before you apply. We do outreach, such as this webinar, and you'll see that we are always out on the road and happy to meet with potential applicants at conferences or regional workshops. Funding, obviously, first and foremost. Networking, trying to facilitate connections between you, maybe NIA programs or external resources. And then entrepreneurship. Several entrepreneurship training programs are now available for our grantees. Next slide.
Slide. The SBIR and STTR program at NIA Innovative Solutions to Significant Unmet Clinical Need. Solutions that have significant commercial potential. Projects that leverage the expertise of the company founder, seek funding to produce feasibility data, Phase 1, or conduct product-focused development activities, Phase 2. Special research interests in ADDF. NIA's SBIR and STTR programs have funding available up to $450,000 for Phase 1 and $2.5 million for Phase 2 in ADDF topics. NIA seeks ADDF research focusing on prevention, diagnosis, treatment, care and caregiving, research and clinical tools, analytical tools, digital health, novel devices. A bubble contains text reading, available opportunities. Omnibus funding, investigation initiated, standard application due dates, January 5, April 5, and September 5. NIA-focused funding, focused priority areas in aging, includes higher ADDF budgets. Application due dates vary.
So at the NIA, our mission area is actually quite broad. We look to fund technologies that will help enable and extend healthy aging. That really can cover a wide variety of areas. In terms of SBIR and STTR, we're looking for things that will solve an unmet clinical need and have significant commercial potential. We do have priority areas around Alzheimer's disease due to increased congressional funding in that specific area, but very open in terms of what types of things for Alzheimer's disease we would be interested in, whether they would be prevention, therapeutics, diagnostics, digital health, caregiver coordination, everything really that can help in the AD space. Many of our applications come through the omnibus solicitation, which is investigator-initiated, but then we also have that Alzheimer's funding opportunity, PAS-19-316, that is specific to anything relating to Alzheimer's. Next slide.
Slide. Priority areas in the National Institute on Aging are covered by its 4 main divisions. Division of Aging Biology. The overall goal is to provide a basis in basic biology for preventative and interventional strategies to increase resilience and extend healthy aging. Division of Behavioral and Social Research. Supports research and research training on the processes of aging at both the individual and societal level. Division of Geriatrics and Clinical Gerontology. Supports research on health disease in older people and research on aging over the human lifespan, including its relationships to health outcomes. Division of Neuroscience. Supports research to further the understanding of neural and behavioral processes associated with the aging brain. Research on dementias of old age, in particular Alzheimer's disease, is one of the division's highest priorities. A pie chart titled Portfolio Classification AD and Non-AD. In vitro diagnostics, 7%. Imaging devices, 5%. Research tools, 9%. Treatments, 37%. Sensors and monitoring technologies, 18%. Supportive devices, 10%. Digital and mobile health, 19%.
At the NIA, we have 4 main divisions: the Division of Aging Biology, the Division of Behavioral and Social Research, the Division of Geriatrics and Clinical Gerontology, and the Division of Neuroscience. So many on the phone are interested in Alzheimer's disease drugs and diagnostics. Those would fall under the Division of Neuroscience. And my colleague, Dr. Zane Martin, will be here for the Q&A, and she represents the Division of Neuroscience as their small business lead. Next slide.
Slide. A table lists funding opportunities by type for SBIR and STTR. Under Omnibus FOAs for SBIR, there are PA 19272, clinical trials not allowed, and PA 19273, clinical trials required. Under ADDRD-focused FOAs for SBIR, There are PAS 19316, Advancing Research on ADDRD, PAR 18512, Lifespan Health Span Extending Interventions for ADDRD Patients, PAR 18329, Technologies for Detecting, Monitoring, and Assessing Cognitive Function in ADDRD Patients, PAR 17067, Tools for Clinical Management of ADHD and its Comorbidities, PAR 18588, Assistive Technologies for ADDRD Patients and their Caregivers, and PAR 18186, Development of Socially Assistive Robots for ADDRD Patients and Caregivers. Under Omnibus FOAs for STTR, there are PA 19270, Clinical Trials Not Allowed, and PA 19271, Clinical Trials Required. Under ADDRD-focused FOAs for STTR, there are PAS 19317, Advancing Research on ADDRD, PAR 18514, Lifespan Health Span Extending Interventions for ADDRD Patients, PAR 18326, Technologies for Detecting, Monitoring, and Assessing Cognitive Function in ADDRD Patients, PAR 17066, Tools for Clinical Management of ADHD and its Comorbidities, PAR 18587, Assistive Technologies for ADDRD Patients and their Caregivers, and PAR 18185, Development of Socially Assistive Robots for ADDRD Patients and Caregivers.
So here's a list of all the funding opportunities that we have. You see the Omnibus, and you also see that PAS-19-316 and 317, SBIR and STTR respectively. Those are the main ones that you would likely apply to. Next slide.
Slide. Already have a Phase 2? Consider the Commercialization Readiness Pilot Program, CRP. Can be simultaneous, or follow on to Phase 2s and Phase 2Bs, both SBIR and STTR. SB1 mechanism enables an absence of subcontracting restriction. The subcontracting plan must still be justified in the application. Special review criteria including a focus of innovation on the product. Provides funding for activities that are not typically supported by research grants.
I mentioned the CRP for those that already have a Phase 2. It's really useful for companies that need to do work that might not be considered traditional R&D and maybe not innovative in and of itself, but critical to advance towards commercialization. And those are things that we could pay for with a CRP award. You can apply either during your Phase 2 or after your Phase 2. Next slide.
Slide. Remember, focus on the product. A diagram on the left is titled Academic Grant and features a hexagon labeled Science. 3 arrows point towards the hexagon with the labels Environment, Approach, Appropriate for Discovery, and Expertise Team Discovery Research. 2 arrows point away from the hexagon, labeled Significance, Knowledge Increase, and Innovation, what could be theoretically possible. An arrow pointing to and from the hexagon is labeled Product, Optional. A diagram on the right is titled SBIR STTR Grant and features a hexagon labeled Product. 3 areas point towards the hexagon, labeled Environment, Approach, Product Development, and Expertise Team, Phase 1, Research and Development, and Phase 2, Commercial Development. 2 arrows point away from the hexagon and are labeled Significance, Changing of Paradigm, and Innovation, Competitive Advantage. 2 arrows point towards and away from the hexagon labeled Science, Mandatory, and Commercialization. The word commercialization is printed in red.
So the SBIR program is similar in many ways to the other research funding mechanisms at the NIH, but the one major difference is the focus on the product instead of the science, but the science is of course underpinning that product and that focus. So as an example, it will have many of the same review criteria, but significance for an R1 will be in terms of how much knowledge are you adding to the field, what's the likelihood of a landmark publication. But for SBIR, significance will be defined as, what is the value proposition? How strong is that competitive advantage? What's the unmet need? So you're still looking at significance in both cases, but really looking at it through a very different lens. Next slide.
Slide. Compose of specific aims page. Specific aims, 1 page, the executive summary and first impression first 1/2 to 2/3. The elevator pitch. Why is it the most meritorious? 1. The technology prototype to be developed. 2. The technical innovation the development would represent, the unmet need being addressed, and the technical challenges to overcome. The value proposition and competition. Include textual highlights of preliminary data and the relevance of the R&D to NIA's mission. Last 1/3 to 1/2. Describe the aims for the proposed project. Include key models, assays, and metrics. Include quantitative performance milestones. Provide your draft-specific aims page to NIA staff for feedback.
I mentioned how we are happy to provide guidance to applicants before they apply. I mentioned earlier that there are a lot of key benefits to the program. In my mind, there's really only one cost of applying for the program, and that's time, because it is a federal program. There are, robust applications that you have to go through and complete. But we are here to kind of demystify that process for you and answer any questions you may have. We suggest that you first develop the specific aims page, which is a key score driving page of the application, and send that to us at least 4 weeks ahead of the application due date. If you do that, it enables us to really provide much more concrete guidance, because many of your questions will be, well, what's the chance that this will be competitive? Is this the right types of experiments to do in a phase 1, a phase 2? Should I do a phase 1? Should I do a fast track? What's allowed in terms of budget? So all of those questions can be answered once we have that specific aims page. A truly competitive specific aims page is so much more than the aims. Really, the specific aims page should be the executive summary of the application and should answer the question to the NIH that if we get 1,000 applications, which we get over 1,000 applications, why would this application be the one that we should prioritize the most for funding? And you should be able to do that in that first half to 2/3 of the page, the elevator pitch. That can include textual highlights of the preliminary data that you would show in the rest of the application, the value proposition, and the relevance. In that last third to half a page will be the aims and milestones that will be part of the project you are requesting funding for. You can send that directly to our office 4 weeks before the due date or more, and we can schedule a call to provide guidance. Next slide.
Slide. Resources to help research entrepreneurs. First-time and never-funded applicants. Applicant Assistance Program. A 10-week coaching program to help develop your application. Offered once each standard funding period. All awardees. Technical assistance budget allowance. Support services such as access to technologies, IP protections, and market research. Offers up to $6,500 for Phase 1 and up to $50,000 for Phase 2 when requested in the grant application. Diversity Supplement. Helps increase the diversity of the research workforce by supporting students, postdocs, and eligible investigators from underrepresented groups. C3I program for NIH. Supports medical device innovators in commercializing their products through a 24-week entrepreneurial training course. Seed Resources. Support from the NIH Small Business Entrepreneurial Education and Development (SEED) office, including access to entrepreneurs in residence and regulatory support. Phase 1 awardees. I-Core at NIH. An 8-week intensive course that offers real-world, hands-on entrepreneurship training in customer discovery in life sciences and biotechnology.
Next slide. So we understand that probably the most important thing we can do is provide you the funding you need to collect the data that is required to attract external investors, including the ADDF, which is a great potential resource for you, and you'll hear more about. But we also have several other non-financial resources to help you. There is the Applicant Assistance Program, which actually had a due date today, but we run it every few months, so there'll be another due date 3-4 months from now. It's a 10-week coaching program to help develop your application. And that's specifically for applicants that have never received SBIR funding before. We also, once you receive your money, your grant, we have several assistance programs. In your application, you can actually request for technical assistance funding, $6,500 in Phase 1 and $50,000 in Phase 2, to provide funds for things like a regulatory consultant or reimbursement consultant or IP, anything of that sort. Once you have your grant, you can apply for several programs, including I-Core, which is an entrepreneurial training program for Phase 1, the C3I, which is a more long-term and expansive entrepreneurial training program specifically for device companies, diversity supplements to expand diversity on your team, and several resources through the new NIH Small Business Entrepreneurial Education and Development Office, including entrepreneurs in residence that are past investors that can advise you on, discussions with external investors and things of that sort, and regulatory support as well. Next slide.
Connect with NIA. Visit https://www.nia.nih.gov. Second SBIR. View upcoming events and funding opportunities. Visit the NIH Grants Guide to search all active NIA funding opportunities. Subscribe to our blog to stay up to date with the latest from NIA. https://www.nia.nih.gov. Research blog. Questions? abusiness@mail.nih.gov. Follow us on Twitter @NIA_SBIR.
So please feel free to connect with us in the future. The one thing I didn't say, which I did want to add in because it's a question we always get, is in terms of the budget. So as I said, 2/3 of the budget has to be going to the company, but that can include fee-for-service activities. So if you're sending something out to a commercially available resource, let's say a CRO, and they have a screen that they sell it commercially, you send them your drug, they run it through the screen, they send you back the data, you do all the analysis. They're billing on a fee per basis, meaning they're not charging any indirect rates. That can count as part of the small business's cost and within the 2/3, because you're essentially buying that service as a supply, and it can be part of your materials and supplies budget. It can't make up all the costs. You still need to be doing some of the work yourselves. But things like academic partnerships, where there will be intellectual input from the academic partner, and they will likely charge indirect rates. Those are subcontracts, and those have to be within the 1/3 of the budget for a phase 1 SBIR, as an example. The last thing I want to say is that please feel free to email us, but if you'd like to meet, we have an upcoming event section on our website, and we do attend conferences around the country. We will actually be in San Francisco for JP Morgan Week, and we have space and staff available, including Dr. Zane Martin, to meet with potential applicants. So if you will be in San Francisco during JP Morgan Healthcare Week, I think the week of January 12th, please email Lainey, who you'll have her email address at the end of this presentation, at the end of the webinar. And Lainey can schedule time for you to sit down and meet with us that week in San Francisco. Thank you very much, and I look forward to hearing from ADDF. Thank you, Alessio, for being a great partner and doing this together.
Slide. Alzheimer's Drug Discovery Foundation. Funding opportunities. To accelerate drug development for Alzheimer's and related dementias, Alessio Travaglia, Ph.D., Scientific Program Officer.
Thank you, Todd. So today I will discuss our funding opportunities and also what we're looking for in a successful application. Next. Over the past 20 years, the Alzheimer's Drug Discovery Foundation, ADDF, has invested over $130 million, and with it funded over 600 programs in 19 countries. So now we have over 130 active programs, and this includes 32 active clinical trials. Next.
Slide. ADDF follows a venture philanthropy model. There are two columns, organization type and investment type. An arrow points left to right from organization type academic nonprofit to investment type royalties. Another arrow points from organization type biotechnology for-profit to 4 investment types, including royalties, equity purchase, convertible promissory note, and warrant.
So we do follow a venture philanthropy model. This means that we make investments. And this typically with the academic is done with royalties, whereas the investment type for companies is royalty or equity or convertible notes with warrants. Now we do way more than funding, because in addition to providing financial support, we can also help to spin out companies, we can introduce to other investors, we can provide validation and also foster collaboration. And something that we do quite often is also helping design studies and providing resources within our network. So you can see in the next slide.
Slide. Diverse targets. Neuroinflammation, proteostasis tau TDP43, neuroprotection, synaptic health and dysfunction, mitochondria and metabolic function, oxidative stress, epigenetics, vascular.
We are target agnostic, and we do have a pretty diverse drug target portfolio. We do support projects in neuroinflammation, neuroprotection, neurotransmission, epigenetic, vascular, and next.
Slide. ADDF funds drug development, clinical trials, and biomarker development. Drug development, translating knowledge into drugs. Biomarker development, validation of neuroimaging and fluid biomarkers. Regulatory studies, preparing drugs for testing in humans. Clinical trials, human clinical trials.
Slide. And to differentiate ourselves, in addition to have a pretty diverse drug target portfolio, we also differentiate our portfolio in terms of key areas of focus. We do support drug development through preclinical studies. We do support development and validation of plasma, CSF, neuroimaging, and digital biomarkers. We do support regulatory studies, and we do support, and we are very interested in clinical trials and epidemiological studies. So,
slide. Next. Core funding opportunities. Eligibility. The ADDF provides funding to academic centers and biotech's worldview. There are two columns, award and duration. Under Drug Development RFP, it's $150,000 to $600,000, 1 to 2 years. For programs to accelerate clinical trials, PACT, RFP, it's up to $3 million and varies. For neuroimaging and CSF biomarker development RFP, it is $150,000 to $600,000 and 1 to 2 years. For prevention beyond the pipeline RPF, the award and duration vary.
Let's look at our core funding opportunities. So the first good news is that, as you can see, pretty much everyone is eligible to apply because we do support academics, we do support biotech, and there is no geographic restriction. We do have 4 cycles over the year, and we do have 4 core RFPs that are listed here, and there are also links, and at the end you're going to have this slide, so you're going to easily have information, detailed information about these RFPs. So what I'm going to do next is to discuss briefly each RFP and also what they're looking for in successful applications. Next
slide, Drug Development RFP. This RFP supports preclinical PK, PD, and target engagement, and in vivo efficacy or proof of concept studies. Novel, repurposed and repositioned drugs, small molecules and biologics, are appropriate. This RFP does not support basic science, target identification, target validation, assay development, and high-throughput screening. This RFP does not support anti-amyloid approaches or cholinesterase inhibitors.
So the first RFP I'm going to discuss is the Drug Development RFP. This does support preclinical PK/PD, target engagement, and also in vivo efficacy or proof of concept studies. We do support novel, repurposed, and repositioned drugs, and we're interested in small molecules and also biologics. Something that is very important to highlight here is that we are interested in late preclinical studies that most likely will reach IND-enabling studies within 2 years. This means, if we go to the next bullet points, that we have very limited interest, and we do not support basic science, target identification, target validation, and screenings. Something else that I need to mention here is that though we are target agnostic, in this RFP we do not support anti-amyloid approaches and cholinesterase inhibitors. So why projects in the drug development RFP are rejected? Typically it's because of the stage of discovery. So again, we're not interested in basic science programs. Also, for programs that aim to test a repurposed or repositioned drug, it has to be really clear why it's important to do additional preclinical studies instead of just going to clinical trial.
Slide. Next. Program to Accelerate Clinical Trials, PACT, RFP. This RFP supports early-stage human clinical trials and regulatory studies. Novel, repurposed, and repositioned drugs, small molecules and biologics, are appropriate. This RFP does not support anti-amyloid approaches or cholinesterase inhibitors.
Next RFP is the Program to Accelerate Clinical Trial. This RFP supports early-stage clinical trials, including phase 0, phase 1, and phase 2 clinical trials, and also regulatory studies, including non-GLP and GLP pharmacology, IND-enabling studies, long-term tox, GMP manufacturing testing. As mentioned before for the Drug Development RFP, we do support novel, repurposed, and repositioned drugs, and we're interested in small molecules and biologics. And as you can see in the next bullet point, again, though we are target agnostic, in this RFP we do not support anti-amyloid approaches and cholinesterase inhibitors. So what's the main reason why proposals are rejected in this RFP? It's typically the lack of biological rationale. So if you submit an application, please be clear why you're proposing this drug, what's the mechanism of action, and also why this drug should work in Alzheimer's disease. Also, typically, the strongest application have a very solid data package, and also are pretty clear in why the dose has been chosen, and also what is or are the biomarkers to measure target engagement.
Slide. Neuroimaging and CSF biomarker development. This RFP focuses on developing novel PET ligands for clinics use, supporting novel CSF biomarkers, validating established MRI approaches in larger cohorts. Peripheral biofluids and digital approaches are supported through the Diagnostics Accelerator RFP. The ADDF has limited interest in CSF measures of amyloid and tau.
Next RFP is the Neuroimaging and CSF biomarker development. This RFP focuses on developing our novel PET ligands, supporting novel CSF biomarkers, and validating established MRI approaches in larger cohorts. Something that I'm going to mention in the next slide is that we do support through an additional RFP peripheral biofluids and digital biomarkers that so are not fundable in this RFP. The main reason why proposals are declined in this RFP is typically lack of preliminary data. And again, remember that we do not support exploratory and basic science projects. And the strongest proposals are the ones that not only have good preliminary data, but also have a very clear context of use, and also clarify how the biomarker compares to other available biomarkers. And last thing I want to mention here, as you see in the bullet point, that in this RFP, we have very limited interest in CSF measures of amyloid and tau. And again, this is because it has to be clear how the biomarker compare with what's already available. In this case, amyloid and tau. Next.
Slide. Prevention beyond the pipeline. This RFP supports prevention clinical trials, comparative effectiveness research, and epidemiological studies.
The prevention beyond the pipeline RFP supports the clinical trial for Alzheimer, but also has conditions that have been linked to increased risk of dementia, including cognitive aging, postoperative delirium, traumatic brain injury. And specifically, this RFP supports prevention clinical trials, comparative effectiveness research, and also epidemiological studies. Next.
Slide. SBIR STTR bridge funding and ADDF NIA and NINDS funding opportunities. 1. ADDF SBIR STTR. Bridge funding to generation additional preliminary data. 2. Scored but not funded. Academic group and biotech companies scored but not funded. The proposed work should fall within the ADDF's mission and current funding priorities.
As Todd briefly mentioned, I'm very excited to announce this new opportunity. This is to provide bridge funding to support companies that have been already awarded with SBIR funding. The goal of this funding is to generate bridge funding to give you the opportunity to generate new preliminary data before applying to larger NIH funding. And I do encourage you all, if you already had a SBIR grant or as soon as you get the SBIR grant, to get in contact with us so that you can know your program and see whether you are a good fit for our funding opportunities. Something else that we do consider are programs that have been scored but not funded by NIA and NINDS grants. And for this case, we have a facilitated way to review your proposal. You will just need to submit to us your review sent to the NIH in NIH format and the comments from the reviewers. And something that I want to clarify here is that, again, all the proposals need to fit within our current funding opportunities and priorities. Next.
Slide. Special RFPs. There are 4 columns: Partner, RFP, Award, and Duration. The Harrington Discovery Institute. ADDF Harrington Scholar Program, up to $600,000, 2 years. The Association for Frontotemporal Dementias, ADDF AFTD Drug Discovery RFP, up to $150,000, 1 to 2 years. The Association for Frontotemporal Dementias, ADDF AFTD Treat FTD Fund, up to $2 million, 1 to 3 years.
We do have several other RFPs over the years, and those include the partnership with the Harrington Discovery Institute and the partnership with the FTD to discover new drug and run clinical trial for frontotemporal dementia. So I would just invite you to check our website frequently for updates on special RFP or sign up to our newsletter to be updated on these RFPs. And next.
Slide. Diagnostics Accelerator. Fast-tracking better tests for Alzheimer's.
Last year, we did announce a novel RFP that is the Diagnostic Accelerator. This is a nearly $50 million funds from partners including Bill Gates, Leonard Lauder, Dolby Family, Schwab Foundation, and Jeff Bezos. And the goal here is to develop novel biomarkers for Alzheimer's disease and related dementias. Next.
Slide. Reliable, affordable, and accessible biomarkers can revolutionize how researchers understand how the disease progresses, identify people for clinical trials, and monitor responses to treatments.
Why we need to develop novel biomarkers? We do believe that having affordable and reliable biomarkers will really change the way we do Alzheimer's disease research, because this is going to give us the opportunity to track disease progression, identify people. Still focus on the previous slide, that have identified people for more efficient clinical trial and allow to monitor response to treatment. Right now we are accepting application on a rolling basis for peripheral biomarker, and here specifically we're interested in biomarkers in blood, saliva, urine, ocular biomarkers, and other. And also we do accept application for digital biomarkers, focusing on symptoms domain including cognition, speech, motor, sleep, and other. And this can be done using digital platforms such as portable sensors or softwares. So again, check our website. We're accepting application on a rolling basis. Next.
Slide. ADDF application process. Before applying, 1. Find the right RFP. 2. Review the evaluation criteria. 3. We encourage to reach out ADDF staff. Next LOI deadline, core and NIH program. January 17, 2020. From top to bottom, a list reads, letter of intent, review of ADDF staff, invited full proposal, scientific and business review board, award decision.
And last, I want to encourage you all to find the right RFP that fits your program. As mentioned before, the main reasons why programs are rejected typically is that do not fit within our priorities. And so please look carefully also our evaluation criteria that are specific for each RFP. And this is really going to make your proposal stronger if you follow the guidelines that we stated. Last thing that I want to mention is that to reach out to us, not only if you have questions, but also if you have any interesting idea that you would like to discuss to us and develop. As I mentioned before, we do more than funding, and we do have also an iterative review process. So we can help you to find resources for your study or run a clinical trial if we are interested in your approach. Our review process starts with a letter of intent, and next deadline is going to be January 17. And then invited full proposals are sent out for external review. And overall, we try to make our funding decision in about 5 months. And in the next slide, you are going to find my contact information. And with this, I want to thank you all for your attention, and I'm happy to take any questions.
Slide. Alzheimer's Drug Discovery Foundation. www.alzdiscovery.org/RFPs. Alessio Travaglia, Ph.D. Email, 8travaglia@alzdiscovery.org. Phone, 212-901-7996. Slide. Questions. To submit a question for either speaker, please select the chat icon from your menu bar and type your question in the box. The moderator will read your question and direct it to the correct speaker. Please note, similar questions may be combined.
Thank you, Dr. Travaglia. We will now be transitioning to the Q&A portion of our session here today. To submit a question for either speaker, That will be Dr. Zane Martin, as well as Dr. Alessio Travaglia. You can please look at the chat feature or the Q&A feature at the bottom of your screen. We have received a few questions already, and so we'll be moving first with some of those. We did get some basic housekeeping questions. One, we've been asked if the slides will be shared, and we wanted to let you know that we will be posting these slides to the NIA website, and we'll be sending an email out about that, as well as the resources that have been shared from ADDF, as well as the NIA Office of Small Business Research. With that said, I'm going to turn the first question to Dr. Martin. There was a question about the commercialization readiness program, and what are the milestones one needs to accomplish and apply for this?
Hi, yes, that's a good question. This is Zane Martin. So the commercialization readiness pilot program is for technical assistance in later stage R&D. One of the requirements to apply for the CRP is that you must be in or have completed a phase 2 or phase 2B grant. For further milestone or to feel that you are prepared to apply for a CRP, I recommend contacting your program officer to talk about what milestones and goals that you have met in your phase 2 and to see the appropriateness and readiness for the CRP program.
Thank you. And I think the next question applies to both ADDF as well as NIA. How strong is the requirement for preliminary data to be provided in the proposal for application? I'll start off maybe if you can go first, Dr. Travaglia.
Sure. It really depends from the specific RFP. But I would say that the preliminary data have to convince us and our reviewer that the program has high success to work. So we don't want something that is based on possibility or review literature. We want, for example, if the drug needs to be tested in a clinical trial, has this been tested in animal model? Do we have information about the safety of the drug, how the dose has been selected? So it really depends on the specific RFP, and so I would encourage the person to ask this question to elaborate more so you provide more detail. But again, like the goal is to convince us and the reviewer that the program has high success to work.
Thank you. And Dr. Martin, did you want to add anything there for NIA specific data to be provided?
Okay, so for phase 1 applications, preliminary data is not required. However, it is very helpful to demonstrate the likelihood of success for a phase 1 feasibility study. Phase 2, there should be preliminary data for phase 2. Preliminary data can include published studies from others in the field that lay the groundwork for the proposed study. And of course, also studies that are done in the lab by that PI. But importantly, the reviewers, for the publications, reviewers rarely read the reference papers. So make sure that all the key preliminary data is shown in the body of the research strategy.
Fantastic. Thank you very much. And there is a follow-up question about preliminary data. The question is, what would be better to include repurposable drug data for patient population, validation data such as insurance claims and EHRs, or the 80 genetic mice model data?
Okay, so I guess I'll answer that first. So from what I'm getting from that question is what is more translatable, which data would be more translatable, because especially in the Alzheimer's field, preclinical animal studies is around 99% don't translate into human clinical trials. So that has been a problem when looking at how best to translate drugs into the clinic. With that in mind, it is good to also have some sort of human studies as well. That could be postmortem tissue experiments for repurposable drugs, as the question was asked, could be valid as well, depending on the purpose of the study. But I think the bottom line is that the studies need to be as robust and rigorous as possible. Alessio, do you have anything else to add on that?
Sure, that's a good question. I could make a specific example of a program that we did fund. The program was to repurpose rotigotine, that is a dopamine agonist used in Parkinson, and to use this transdermal patch of rotigotine in Alzheimer patients. And so in this case, the evidence not only were coming from the Parkinson field that the drug was safe, but there was an introduction discussing why do we have to target dopamine in Alzheimer patients. So what's the expression level of relevant receptors and downstream mechanism in the Alzheimer brain? Is this something that is downregulated? It's convincing the fact that if we rescue this mechanism, it's going to bring a benefit. And the other thing that we do want to see in a case like this is also how are we going to measure this. In clinical trial application, we do not want to see cognition as a primary outcome. We ideally would like to see a biomarker that is going to give confidence that the drug is working the way it's supposed to work. And also is going to give confidence to other investors to further support the program.
Okay. Well, I'll give our speakers a brief minute to catch up. I wanted to share some quick reminders. You can submit questions through the chat and Q&A feature at the bottom of your screen. In the chat, we have actually included some links to the ADDF as well as the NIA small business website, so you can look at some of the contacts we mentioned and the open funding opportunities that are available right now. With that said, I'll turn to our next question. Is there a specific program for women-owned companies or for PIs from underrepresented groups? So essentially, is there any advantage to support these underrepresented groups through either ADDF or the NIA SBIR program? I'm going to start off with Zane on this question.
Okay, sure. So we, NIA has just joined with NCI, the National Cancer Institute, on the Application Assistance Program. That's our main program that caters to women-owned and other underrepresented groups. There's also local programs for those in IDEA states, which we can point you to. You can send us an email, any of your program officers an email with that question.
Great. And I was also going to note that the institutes at NIH do track and look at who is applying. So if you do apply, please do select whether you do have a woman minority owned or if you're from an IDEA state on your application, as well as the Small Business Administration has a variety of resources to help women and minority owned small businesses. And with that, Alessio, I'll turn it over to you to see if you have anything to add there for ADDF.
Well, the last thing that I want to add is, as mentioned before, if you have any questions, if you don't know if your program fits, or if you have an idea but you are not sure about how to develop it, please get in contact with us because we're happy to talk with you and we're happy to help if we can.
Great, thank you. Another question targeted for the NIA funding opportunities. There's a question about a small business, whether they should be using the NIH negotiated indirect rate, the minus rate of 10%, or should they budget out of their anticipated F&A not to exceed 40%?
So for indirect rates, you can request up to 40% without having to give justification for that.
Thank you. One other question, and if anyone else has questions, please do send them through. But for either program, do the funds have to be repaid back, and are they do they lose any IP control? I'll start off with ADDF.
So we're talking about companies. We do sometimes convertible notes with warrants or equity, and yes, there is something that we need to receive back. And again, the goal is to receive capital that we can invest in other research. In terms of IP, we absolutely do not have IP.
Thank you. And Dr. Martin, do you want to talk about what the benefits are through NIA?
Sure. Through NIH, it's a non-dilutive program, so there is no repayment that's required. And the intellectual property rights are retained by the small business.
Thank you. I will pause to see if I see a couple other questions coming through. I just wanted to note at the conclusion of this webinar, you'll be getting some feedback to fill out. If you would please fill out the link and share with us any particular feedback or things that you'd like to see in the future. We do take that feedback very, very seriously and adjust as well as respond to your needs. The next question is coming and asking about models for demonstrating efficacy of a new therapeutic agent, given that neither mice nor in vitro models are considered representative of the human disease. Dr. Martin, I know you were just touching on that earlier. Do you want to expand at all?
Sorry, I didn't see that question. Can you repeat that one?
Yeah. Yes. So what do you think about models for demonstrating efficacy of a new therapeutic agent, given that neither mice nor in vitro models are considered representative of the human disease?
So that's a great question. It's one of the biggest questions in the field. The main goal for preclinical is you want to demonstrate target engagement. So that, of course, can be in vitro or in vivo. So the model, I mean, while it would be ideal if we had an animal model that perfectly recapitulates Alzheimer's disease, we don't. Another approach that can be taken is to find a model that represents the therapeutic target that person is investigating, so they can investigate target engagement of that model. In target engagement, to move that further, then they need to also show biomarker with that target engagement using biomarkers so they can translate it into human clinical trials.
Next question is, what are the challenges you are facing in human clinical trials? So I will note, and Dr. Martin, perhaps you can share this, but in terms of NIA's involvement and whether we do fund clinical trials, maybe that would be another way to look at that question.
We do have a nicely robust clinical trial program. Half of our clinical trials are pharmacological with the Alzheimer's, in the Alzheimer's area. About half of our clinical trials are pharmacological clinical trials for Alzheimer's therapeutics, and then the other half is non-pharmacological clinical trials. I don't have the exact number, but I can say that in looking at our pharmacological clinical trials, we target many therapeutic target areas, not just amyloid or tau, but also synaptic plasticity, vasculature, oxidative stress, just to name a few.
Great, thank you.
Can I add something about the previous question about how to translate the study in vitro in animal model? I want to add what we do like in applications when the model is not, let's say that the drug target vascular aspect in the brain. The ideal animal model is not necessarily transgenic model of Alzheimer. It has to be a model that is relevant to the mechanism study. So it could be just a model of vascular lesion in the brain, for example. And as Zane said, like we really like to see target engagement in these studies. Something that often is missed is a translatable biomarker, meaning that if the study is done in animal model, you could do Western blot immunohistochemistry, but then how this is going to be done in human clinical trial. So if you have a CSF plasma biomarker or a PET ligand, that's certainly going to strengthen your proposal. And we do look also in these animal studies, we do look already at the potential to run the clinical trial. So even though it's an animal study, we still want to see how the clinical trial is going to be run.
Thank you. There is a question for clarification on ADDF funding and whether the funding is non-dilutive.
No, we do, for academics, we do have royalties, but for companies, we do typically convertible notes with warrants, or sometimes we take equity in the company.
Thank you. And then the NIA funding is non-dilutive.
Correct.
Another question relates to confidentiality in terms of how much data is shared. How confidential is the information during the review process, and would it be necessary to first file a provisional patent application prior to submission of an application? Dr. Martin, will you take that one first, please?
Sure. So the review process is very confidential. The reviewers have to sign a confidentiality agreement before they review. But that said, it is the application would be viewed more highly if they did have a patent application prior to the submission, because it shows greater potential for commercialization. But I do get that question a lot by applicants for proprietary reasons. But to ease those fears, reviewers cannot give any information outside of the study section. I mean, now that said, of course, I'm sure that that's happened before. So one thing I like to advise is if people are very worried about it, in showing, like, for example, a structure of their compound, I advise to give as much information as possible for the reviewer to be able to evaluate the feasibility of that compound, such as the physicochemical properties, any like PK properties, and to show as much of the structure as possible, and then put maybe black boxes over any part of that structure that they don't want to be seen for proprietary reasons.
Thank you.
And for us, we do also keep all the information shared with us confidential. All our reviewers are under CDA, and sometimes companies ask to sign a CDA with them, and we're happy to do so. And I agree with Dane, highly recommend to share as much information as possible to make the reviewer's job easier. Thank
you both. The next question relates to whether there is interest in developments of amyloid-related biomarkers or drugs. Is NIA or ADDF still funding this area?
Okay, so I'll speak first. Sorry, Alessio. We do still fund amyloid-related biomarkers or targeted therapeutics. But we highly encourage an increase in novel targets and novel biomarker discovery. For amyloid related therapeutics, we like to call those next generation amyloid therapeutics because it's not the same as past amyloid therapeutics such as immunotherapy and other secretase inhibitors, stuff like that. So we do fund the next generation amyloid therapeutics. We do that.
And for the ADDF, we do not fund drug development or clinical trial on amyloid related. We do support biomarker for amyloid. Let's say that there is a novel way to measure amyloid in plasma or in saliva or in the eye. This is something that we're certainly very interested to see.
Thank you. I do want to just send a reminder to folks that if you're interested in meeting with NIA staff and you'll be at JP Morgan, we did include in the chat some information about how to schedule that one-on-one in-person meeting. For both ADDF and NIA, you've heard from our leaders that they are open to helping talk to you before you submit an application and discuss your research areas of interest. That is a very important tip in terms of developing a successful application and making sure that where you're applying is a good fit and know that there's an investment of time with each application. With that said, I want to thank you for your time. Again, please do respond to some of the feedback questions we're going to be sending you. And I also want to thank our co-hosts, the National Institute on Aging Office of Small Business Research, as well as the Alzheimer's Drug Discovery Foundation. Thank you all for your time, and we hope that you'll apply for these important programs.
Text, Monique Larocque.